more time now, and I actually look forward to hearing from Professor Lukmanova about the program as she sees it today, and then from the students about their impressions. Maybe I can say a few words, a few comments after I have listened to you all.
Okay. Could you start?
Yeah,
Okay.
of course. And first, I want to express my unlimited happiness that we finally meet and that we all healthy. And we had the opportunity to talk with Kurt Wütrich. This is a great pleasure for me and for all of us. So I want to present a little presentation about our program and our disciplines and our lectures. So please wait. Okay, I start. Okay. On this slide, you can see the three men who are the founders, or I can say that they are parents of our program. This is Professor Kurt Wütrich, who for the first time in 1985 determined the protein structure by NMR, and then he won the Nobel Prize in 2003. And this is Professor Arsenyev, who also in 1985 determined the first structure of peptide membrane channel formed by Agitoxin Ii. And this is, you see, academician Mikhail Kirpichnikov, who also previously worked in NMR, and now he is a dean of biological faculty of Moscow State University. And these three people in 2018 decided to organize our program and wrote special memorandum about the opening of our program. And now I want to say several words about this program that we started in this year. So this is some disciplines, some courses of our program. It is not all, but the main. Of course, we pay big attention to the structure of the proteins and nucleic acids. We talk with students about the structure of cell membrane and lipids, and we discussed usage of distant membrane mimetics in structural biology, because this is very important for study of membrane proteins. Of course, we study NMR, EPR, cryo-EM, X-ray, molecular modeling. Also, we give students different programs and applications for visualization and analysis of special structures of biomolecules. We studied programming on Python. This is the most popular programming language presently. And of course, we pay big attention on protein engineering, because everybody understands that before you start to study the special structure of the biomolecule, you need to develop the recombinant system for production of this protein. And we give not only lectures for our students, but we also have a large course of practical classes devoted to cell-free system for production of different cells from E. coli, yeast, and mammalian and insect cells. So we give our students the most modern techniques used now in structural biology. In this year, this book was published. This is the name of this book in English, Modern Methods for Study of Structure and Function of Ion Channels. And this book was written by the lectures of our program. And this book collects all last aspects of structural biology and structural biology used presently. Okay, and now I want to introduce our lectures. I want to begin from NMR. NMR course is taught by guys from the biggest structural department, NMR lab in Russia, from Shmyakin-Овчинников Institute. There are Professor Захар Шинкарев, Dr. Alexander Paramonov, Dr. Maxim Dubinny, Dr. Konstantin Mineev, and Mikhail Myshkin. The student listen not only lectures, but also attend the practical classes. They learn to assign signals in the mass spectra and determine the special structure on the example of the little peptide. It should be noted that all our lectures are not simple lectures. They all have their own labs, scientific labs, and they perform scientific work. So NMR guys, this is a slide with the main tasks that studied by our NMR lectures. There are voltage-gated ion channels, membrane proteins of type 1, and new low-weight molecules such antibiotics or bioluminescent compounds. Here you can see the photo with the worm who expressed unique bioluminescent system, and compounds of the system was determined by our lectures. So X-ray is taught by Dr. Mikhail Boyko. Here you can see the structure of human butyrylcholinesterase tetramer, an enzyme that is the basis of antidotes for organophosphate poisoning. And Dr. Boyko determined its structure and published recently in Biochemia journal. Cryo is taught by Professor Olga Sokolova and Dr. Tatiana Stanisheva-Konovalova. Here you can see the structure of bacteriophage chaperonin, and they demonstrated for the first time its unique arrangement. This chaperonin contains 3 pairs of subunits and 1 separate subunits, and they demonstrated that binding of nucleotides leads to increase in the conformational mobility of the subunits. This is the last of their work. So computer modeling and programming on Python is taught by Dr. Alexey Shaitan and Dr. Grigory Arbiev. Here you can see the structure of nucleosome, and the guys developed computer and modeling methods to study the structure and dynamics of a number of nucleosomes and their complexes. And you can see that they published in very good journals, for example, in nucleic acid research. This is Professor Alexey Feofanov, who taught optical spectroscopy and confocal microscopy. He developed new techniques for fluorescent microscopy of single nucleosomes and their complexes, and these techniques allowed to obtain new structural information about the arrangement, and this work was published in Nature Structural Molecular Biology. Especially I want to say about the course on electrophysiology. Many labs study ion channels because ion channels are the targets for many drugs and very attractive for scientists and big pharma. So, but before you start to study the special structure or the recombinant protein, you need to confirm the functionality of ion channel, and it is important to have in arms the techniques which allow to study functionality of ion channels, and now we have these techniques in our lab. And Dr. Dmitry Kulbatsky is taught the course of the lectures and practical classes, and he learned the students the physiology on R sites or CHO or IHET cells to study the channel gating of studied proteins. Also, I want to say several words about protein engineering course in our program. As I was mentioned above, it is a very important field in structural biology because before structure determined, you need to obtain the object of your study. And this course is taught by me, Dr. Mikhail Shulepko, and my PhD student Olga Shlepova. We use different expression systems from cell-free to production in mammalian cells and insect cells. You can see the photo of insect cells here, transfected by baculovirus. They are green because they express GFP protein, and these cells express also TRPV1 channel. And this is one of the objects of my interests. And then I want to say several words about the studies in my lab presently carried out. So presently in my lab, we studied 3 membrane proteins. This is alpha-7 nicotinic receptor, acid-sensitive ion channel type 3, and human TRPV1 channel. And this work is carried out in collaboration with Kurchatov Institute. This is institute, the one institute in Russia who has cryo microscope, Titan cryos, you can see on the photo. And we use different expression systems, and I want to say several words about one object. This is our last work devoted to study of alpha-7 nicotinic receptor. This is very important receptor because it participates in different cognitive processes and in cancer development. So we expressed in G cells the extracellular domain of this receptor and obtained the sample with a homogeneous sample. You can see here second analysis of the sample, and then we studied the sample with cryo-EM in Kurchatov Institute. And you can see here the photo of images of alpha-7 domain alone and alpha-7 domain with the toxin VTX. It is important to note that it is not established yet how this non-conventional toxin interact with the receptor. So it is very interesting, but unfortunately you can see here that the receptor demonstrated preferred orientation on the grids. We see mostly top or bottom view, and we have very low amount of side views of the domain. Nevertheless, you can see here that in the sample with the added toxin, additional densities are appeared in the interface of adjacent subunits, and this is the toxin. And we can obtain the cryo structure or the domain with the toxin. In spite of low resolution, we can obtain the stoichiometry of this interaction, and you can see that the toxin interacts with the lower part of the domain. And this observation allowed us to propose that the toxin interact with the membrane environment of the receptor. So we studied this hypothesis by NMR, and indeed, by NMR we confirmed that the toxin has membrane binding site. This is shown in green. In parallel, we studied the residues of the toxin important with interaction with the receptor. They shown by this circle. So we have 2 sites the sites important for binding for receptor, and the site important for binding to the membrane. And this additional data allowed us to build by the computer modeling the good model or the complex or the toxin with the extracellular domain of the receptor, and then we build the complex or the toxin, the wall receptor in the membrane environment. You can see here the side view and top view, and here you can see the movie. We performed the long ND simulation, and you can see that the complex is very stable. This is ND simulation more than 1 microsecond. So at present, this is the best model of this receptor, and now we prepare this data for publication. This is a movie demonstrates the receptor from different sides. Wow. And here I want to present our logo of our program. You can see that the silhouette of Moscow State University, and this is the structure of ion channel renin receptor, very important receptor. This is the year of the establishment of our program. Thank you for attention. Maybe you have some questions.
Thank you.
Kurt, do you have some questions about the program?
Not questions. I think that as far as I remember, this corresponds pretty much or exactly to the outline of the course that we have designed. And it is comprehensive. Maybe it's almost too much rather than too little for the students, but maybe we'll hear from them at some point why they are in this program.
Yeah. Okay. I want to introduce now our students. They talk about themselves and about their work. Okay. Yes. And first, I want to invite Демченко Евгения. Женя. Okay.
Hello, let me share my screen. Can you see it?
How do you pronounce your name?
Evgenia, it's my full name, and Jane, I can go just by Jane to simplify the things. So, hello everybody, I'm delighted to meet all of you here. It's my pleasure to present my work in front of you. My name is Evgenia Demchenko. I used to work in several laboratories. I worked at Igor Plotnikov laboratory, where I focused on the comparison of the fluorescent dye rhodamine 123 metabolism in benign and malignant cells. Then I had an internship at David Cleham's laboratory at Janelia, where I was plunged into structural biology for the first time in my life. And there I was involved in the project on resolving polycystin 2 like 1 ion channel structure. And recently I joined Ekaterina Lukmanova lab, where now I'm a participant of 2 projects that I'm going to talk about today. The first project is connected to a voltage-gated potassium channel shaker subfamily member 3 ion channel. It's widely expressed in neuronal and immune system cells. These voltage-gated potassium channels participate in a great number of important cell processes, for example, in resting potential maintenance and cell volume regulation. Its inhibition is an efficient approach for treatment of autoimmune, oncological, and different metabolic diseases. One of the most efficient blockers of KV channels are toxins from Scorpio venom. The toxins are small proteins that block the passage of potassium ions by binding at the pore domain, here at P loop, on the extracellular side of the channel, thereby inhibiting the ion flux. One of such toxins is Agitoxin Ii. Its structure is determined by NMR. It's a small peptide with an alpha-beta motif and the 3 disulfide bonds. It is known that the toxin's lysine 27 amino group competes with the potassium ions in the selectivity filter, thus preventing the ion flow. Our goal is to determine the exact binding of Agitoxin II to Kv13 channel and possible conformational changes, movements of the toxin in the bound state. To achieve this, we have designed a chimeric ion channel. We took a prokaryotic potassium channel from the soil bacteria Streptomyces lividans, KCSA. The structure is shown here. This channel is widely used as a model of the shaker-related potassium channel in eukaryotes. And then we replaced 15 amino acids from the original P-loop with the amino acids from a P-loop of Kv13 ion channel with one additional mutation. One amino acid was replaced by cysteine that is required for the following BTFA labeling for subsequent completely new for me NMR studies. Then we are going to add a toxin, label it with a spin label and try to determine the exact position and spatial movements of the toxin at the Kv13 P-loop. At this point, we have some results for one of our mutants. The red line is a spectrum for the solitary channel and the blue one for the channel with the toxin. We can identify 3 different states for the toxin. It means that the toxin moves from one position to another. The calculated time for the toxin freeze in one position is about 10 milliseconds, so it changed the state not that fast. Of course, we will continue to work on it to collect more data in order to determine the exact states and structural aspects of toxin binding. The second project is related to acid-sensing ion channels. These are trimeric proton-gated and sodium-selective members of the ENAC channel superfamily. These ion channels, they are expressed throughout vertebrate, central and the peripheral nervous systems. In sensory neurons, they are proposed to mediate the pain induced by acidosis, and they may play a role in mechanoreception and spatial learning. On a broad scale, ASICs are potential drug targets due to their involvement in pathological states such as retinal damage, seizures, and ischemic brain injuries. ASICs are encoded by 5 paralog genes in mammals. The structure of the ASIC1 is resolved and it is shown on this slide, but we are working on the resolving ASIC3 structure using the BAC-MAM expression system and cryo-M. We've already cloned the gene to the PFAS BAC vector and extracted the BAC meat, but of course, the most exciting results will come soon. Thank you for your attention. Do you have any questions? Or we can move to another person.
How long have you been at Janelia Farms?
I was there for 2 months.
With whom did you work? Who was your PI at Janelia Farms?
My supervisor was Raymond Hulse, but of course I talked to David and discussed the project a lot as well.
Okay, Katya, I think we could move ahead. Звук.
Sorry. Yeah. I think that the next will be Philippe. Philippe.
Yes, I'm here. Good evening, everybody. Yes, dear professor. Yeah, I'm sorry, I have forgotten to turn on the demonstration. Yes, just a second. Yes. Just a second. Do you see my demonstration? I'm sorry.
Yes, we do.
Yes, okay. Thank you. Dear Professor Wütrich and dear Professor Kirpichnikov, my name is Filip Gavronsky. I got my bachelor degree in the field of molecular biology, and now I get my master degree in the field of structural biology. So, dear colleagues, let me present you my work made in the laboratory of Professor Murat Yusupov in Institut de Génétique et de Biologie moléculaire et cellulaire, IGBMC, University of Strasbourg, about ribosome crystallization of Staphylococcus aureus. S. aureus is a pathogenic gram-positive bacteria. It has different unique features, like persisting on different surfaces, surviving during long-term exposure to ultraviolet radiation, and a wide range of pH. It's necessary to take into account that about 60% of human population has aureus on skin, hairs, and upper airways. In most cases, the bacteria was found as a reason of abscess, but also brain and lung infection diseases. The main danger for humans is a great number of antibiotic-resistant strains. Some of them are presented on the right side of the screen. Last time, the Yusupov's lab conducts researches of the protein biosynthesis mechanism in pathogenic microorganisms. One of them is S. aureus, which ribosomal structure was resolved by cryo-EM with a resolution 3.8 angstrom and recently with 3.2 angstrom. You are able to see the structure on the left part of the screen. Unfortunately, cryo-EM has some disadvantages. That's why we try to get a crystal structure of this ribosome. So the purpose of my work was to purify Staphylococcus aureus ribosome and to test new crystallization conditions and getting new crystal form of S. aureus ribosome. So first of all, we got a recombinant enzyme, lysostaphin, which is necessary for disrupting of cell wall. We prepared lysis solution, including lysostaphin, DNase first type, DDT, and protease inhibitors, and then ended S. aureus biomass. After that, pH 8K was adjusted to 4.2%, and then we centrifused the sample to precipitate high molecular weight structures as ribosomes. Precipitate was dissolved in buffer A and laid sample on sucrose cushion with high concentration of potassium chloride and some additives of magnesium acetate for stabilization of ribosome structure. Then we centrifuged our sample for 17 hours. This step was necessary for removing hide-wide enzymes, translational factors, and the contamination of mRNA and tRNA. Pellet was dissolved in buffer E. The samples were layered on sucrose gradient and centrifuged during 13 hours. It was necessary for separation of 70S ribosome fraction from 100S, 50S, and 30S fractions. We collected the fractions with peristaltic pump with OD more than 4 units to avoid dilution. Interesting feature of our object is a ribosomal dimer 100S. Most bacterial species have this dimer in an active form of cells, but we took our cells on long phase. Unfortunately, the exact role of this dimerization is unclear. Some researchers are found to take part in antibiotic resistance. Our sample were concentrated by pH 20K and centrifuged. Pellet was dissolved in buffer G. We verified our samples on RNA agarose G electrophoresis and SDS page. For verification of results, we used controlled ribosomes, which were purified earlier. As you may see, there are no contamination of mRNA or tRNA on agarose G electrophoresis. On SDS page, there are only lines for proteins of small and big ribosomal subunits. We prepared new crystallization mixture solution. During robotic and handling screening, we got crystals under different crystallization conditions. Unfortunately, that crystals had a bad diffraction. That's why we continued to work. We decided to change the ammonium thiocyanate to potassium ion due to its structural role in organization of functional important ribosome structures as a decoding center, peptidyl transferase center, and general stabilization of rRNA structure. Also, we found that ammonium ion promoting ribosomes aggregation on crystallization plates. Thiocyanate ion has low level of hydration and may interact with positive charged parts of proteins, but it is not seen on electron density maps of thermophilus ribosomes. So we are considered to think this ion creates right ionic environment for crystallization, increasing or decreasing ribosomes solubility. After that, we used robotic screening for optimization salt concentration. Here you may see these conditions. Then we did handing screening for improving the size and quality of crystals. We prepared new crystallization mixture. Magnesium ion is necessary for RNA stability too. Pitch 8K and 10% MPD creates a hydrophobic environment which promotes creating of nucleation centers. Then we titrated our sample for pitch 8K from 3.7% to 4.8%, moving up for 0.1%. We prepared ribosome samples for crystallization. Deoxybig-CHEP prevents aggregation of proteins during crystallization. Spermidine increases ribosome solubility and the size of ribosomes crystals. Then we made scenting drop vapor diffusion. We did it under two different temperatures, 289 Kelvin and 297 Kelvin. The most successful was 297 Kelvin. But under low temperature, we had more crystals for one well, about 40. But they were smaller. Also, we tested the influence of crystallization volume. The crystal size dramatically decreased under low volume. We found new ribosomal crystal form. It's presented here. We are going to use this form in future researches. That would be all. Thank you for attention.
Thank you, Philippe. Kurt, do you have any questions to Philippe?
Well, again, I am interested to see that you have been abroad. You have been in France.
Yes, I have. Yes, it was my internship in Strasbourg.
Who did you work in France?
Could you repeat, please?
Who was your advisor in Strasbourg?
Yusupov. No, he was my PI, yes, and advisor was Alexander Golubov, but he's a PhD student, so I don't think that you know him, unfortunately. So it's
okay.
Yes.
Who is the head of the department where you have been in Strasbourg?
The head is of the department. It's Bruno Claholz. You know, Bruno.
Yes. Yeah, I know where you have been then.
Yeah, Marat. No, and Marat Yusupov is my PA. Yes, it's the head of the lab, and it's the lab of Marat and Gulnara Yusupova. So it's yes, Yusupov's lab.
Okay.
Well, do you know the work? Do you know the work that has been done by my colleagues in Zurich?
Yeah, I know Bond. Yeah, I know Bond. Yes.
You know Bond?
Yes. It's a great crystallographer. Yes, and he's now working with the cryo-EM. Yes.
Yeah. Oh, yeah. It's mostly cryo-EM. Okay.
I think.
Well, do you now have a cryo-EM facility in Russia? When I visited last, which was just a year ago, there was no high voltage EM in Russia yet. Do you now have access to a?
Is it a question for me or for?
Yeah, this question is for you or for anyone who might know.
We have two cryo microscopes, yes, now in Russia now. So yes, we have this possibility. I think it's, but yes, you know that maybe it's better to have it more, but now it's, but however, it's enough to make work on in the field of structural biology. However,
No, dear Kurt, yeah, I can say several words. Now we have a pity situation with microscopes, cryo microscopes in Russia. We have only one big microscope in Kurchatov Institute. This is the CRYOS. And last year we obtained two smaller microscopes in Moscow State University in biological faculty by Professor Sokolova. Bought this microscope, and we hope that we have now opportunity to perform additional different scientific works on this microscope. And a little cryo microscope was established recently in Moscow Physical Institute, Physical-Technical Institute, Fistech, but it is very little amount of microscope for such big country, and maybe sometimes we have more big microscopes like Titan cryos. We hope. They are very expensive.
Yeah, but you have made progress. I mean, because I think a year ago, none of these instruments had been installed as yet. That's very encouraging.
Yeah. So now we can move, yes. And I want to invite Ekaterina Maslova, who talk us about cryo work. Katya.
Yes, thank you. Good evening, everyone. Do you hear me?
Yeah, we
Yes.
see you, but I didn't see you.
Can we ask Philippe to stop his translation?
I have stopped it.
Yes.
How can I do it? I'm not sure because I'm able to pause. Yeah, here. Okay, yeah.
Thank you. Ekaterina Maslova, and I'm very happy to be here for this meeting. And first of all, I would like to express my gratitude to all the people present here for providing me with the opportunity to study at the program of structural biology and biotechnology, and receive the most essential knowledge for the further work. And my way to structural biology was quite long, and it began with the fact that I studied medicine, and along with it, a large number of interesting disciplines, but they were rather detached from my current kind of activity. So now I'm very glad to finally come with a narrow specialization and immerse myself in my huge interest. So in fact, this program is the only one that can allow it. In a bachelor's degree, and now I'm a member of the scientific group of Professor Olga Sergeyevna Sokolova. In the course of my bachelor thesis, I resolved the structure of the chaperoning of the bacteriophage AR9 according to cryo-electron microscopy data. And let me remind you what chaperonings are. They are protein complexes that contribute to correct folding of polypeptide chains. And at the moment, there are two large groups of chaperonings. Group 1 includes chaperonings of bacteria, mitochondria, and chloroplasts, and group 2 chaperonings of archaea and cytosolic chaperonings of eukaryotes. And this particular object is interesting in that not so many viral chaperonings are known in total. There are only 3. And the first 3-dimensional reconstructions were obtained for bacteriophage EL, you can see it, and OBP bacteriophage viral chaperonings. And the structure of the last was also obtained in our laboratory. the data obtained in the course of previous studies suggested that viral chaperonins can be isolated into a separate group, but they still not studied enough. In the course of my work, there was a three-dimensional model of the AR9 bacteriophage chaperonin with a C1 symmetry and with a maximum resolution of 4.1 angstroms. And then the PDB structure was inserted into it. For the studied chaperonin was shown a similarity in the arrangement of alpha helicals with respect to the well-known and well-studied bacterial chaperonin, GROW-EL. Currently, work is underway to obtain the structure of the bacteriophage chaperonin in connection with its substrate and lysine. And in the future, I plan to choose more complex objects like membrane proteins to work with. And I wish to expand the area of my practice to wet laboratory. And I want to work more with methods of releasing, isolating, and purifying of proteins. So that's all. And thank you for your attention. Maybe you have some questions.
Thank you.
Which techniques do you plan to use for studying the binding of substrate proteins?
Single particle CRU-AM. So I just get data from my supervisor and I work with it in special programs. So
You do not perform measurements yourself.
no, unfortunately.
But when you continue now, so you are actually working with computers mainly.
Yes.
Okay.
Only.
Okay. Now we move to Daniil.
Okay, yes, I'm here. Do you see my presentation?
Yes, everything's fine.
Okay, thank you. I will start. Hello, my name is Daniil. I am very glad to see you here. I have finished bioengineering department, biological faculty of Moscow State University. I decided to join structural biology and biotechnology program, master program, because I think it is very interesting field of science, where I can study a lot of powerful methods such as NMR, cryo-EM, X-ray, some methods from wet biology, and which is most interesting for me, computational biology. I think that now scientific community have more data than we can process. So I think now we can get pretty results, just have computer and internet. I'm going to tell you something about work on peptide CM24. It is the smallest peptide which can affect inactivation of sodium channels, doesn't have disulfides, not previously described. There is mutagenesis data. It is important to figure out how this toxin affects on sodium channels of insects, so we can produce some selective insecticides based on this peptide. BGNAF was taken as a model channel, but its structure still unknown, and effect on inactivation indicates a possible interaction between CM24 and force voltage sensitive domain. Peptide CM24 and its mutants have disordered stage in aqueous solution, but adopts an alpha chemical conformation in the presence of micelles. Here you can see cyclar decreasing data, where you can see that this peptide have disordered conformation in water and become alpha helical in detergents. Also, you can see here a structure of this peptide in presence of micelles. This structure was resolved by using NMR, and also you can see mutagenesis data, where you can notice one very important residue, phenylalanine 7s. If we change it with alanine, this peptide stop interact with channel, sodium channel. Also, a complex model of CM24 with a force BG-NAF domain obtained by protein-protein docking. Homology model of BG-NAF channel was constructed using template of cryo-EM structure of NAF path channel from American cockroach. And during this work, we have two objectives: develop an expression system for force BG-NAF domain and study structure of complex BG-NAF domain 4 with CM24 peptide using NMR. Thank you for your attention. You're welcome to answer some questions if you want.
Where will you perform your NMR studies?
This work performed in a laboratory of Zakhar Shenkarev in Ovchinnikov and Shemya.
The 4 students that have presented today are working on very difficult and ambitious projects. And it seems to me that there is not much biotechnology. It is mostly structural biology. Do I see this correctly? Or what will be the how much of the you see, the theme of the program is not only structural biology, but it is also bioengineering in some ways. And I don't see any of this in the projects of the 4 students.
If we got a crystal structure of ribosome, it would be easier to test some antibiotics. So it's a biotechnology, I think.
I see. That's where you see biotechnology.
Yeah,
Okay.
yeah. I don't think so, because in the project of Evgenia Demchenko, that she talked about NMR study of the chimeric channel of KCSA with KV13. She needs to express these chimeric channels in Nikolai systems, and after that you need to express the toxin, also Nikolai cells, and then you need to label the toxin with a magnetic label and the channel with the Phtor atoms, and only after that she will start to perform NMR methods and NMR work. In the case with ASIC 3 channel, Zhenya need to develop from the zero the expression system for this channel in insect cells or mammalian cells. This is the big part of the biotechnology work. In the case of Daniil, also the part of the work is devoted to biotechnology, because the domain of the receptor that he want to study by NMR should be expressed by cell-free system. So the most of our students need to perform some biotechnology work, and after when they obtained the object of their own study, they will start the study of this object by different structural techniques.
Okay. Well, I also want to commend the students for their good knowledge of the English language.
Yeah.
I'm leading a group of graduate students in China. And if they were speaking English as well as your students do, it would be much easier for me.
Yeah. Okay. Maybe some words has Professor Kirpichnikov, Mikhail Petrovich.
Yeah, of course. It was very interesting for me. I agree with Kurt that the most of the work, it is structural biology. As concerned wet biology, it is not real biotechnology, it's wet biology only. But I think this is our first experience, and I think in future we will have some works in real biotechnology with some practical results for it. Of course, using the approach of structural biology. Good, I think we should, and I'm sure that we will continue to distribute the information about our program. And I should say that reports on our program have already been presented at several student schools. And next year we plan to open the possibility of recruiting of foreign students for our program. We believe to introduce some kind of reward for best studies. Perhaps it will be either an opportunity to pay for participation in scientific conferences, or maybe the introduction of some kind of supplement to the scholarship. Also, we hope the next year the competition for the program will be even higher than this year. And students from other universities, for example, Moscow Institute of Physics and Technology, so-called FISTER, will join the graduates of the Faculty of Biology of Moscow State University. I'm sure that our program will be success next in the future. And I would like to say thank you for Professor Lukmanova for this Zoom meeting, of course. And I would prefer to have such meetings maybe once in 2 or 3 months, at least. It would be very useful for us. What do you think about it?
Well, that's certainly a possibility. Sure, you just let me know when you see a good date for next meeting. By the way, are you all at the university now, or are you at home?
I at the university. I am at the university.
Yeah, we are not supposed to be at the university anymore. They ask us to do home office.
Unfortunately,
Yeah.
today I should be at the university today. Sometimes I'm working at home, but today at my office.
Okay. Are the students are also in the university now?
I don't know. I think you mean our
Yeah,
students?
do you give courses in the university?
Till now we have some courses at the university and some courses at online.
Some are online. I just see you are wearing a mask. I also have a mask, but because I'm in home office, I don't wear it.
Okay. What about Zurich? What about ETH? You have some lectures at the university, at the institute, or all studies online?
All big lectures with more than 50 students are not in person. Only small groups of students are taught in personal contact.
Yeah.
And the situation has been very good. There was almost no COVID here in the summer. And during the last 3 weeks, it has simply exploded. At this point, we have 22, I mean, yesterday they had 22% of positive tests. And the situation, statistically, the situation is now worse than in the United States, for example. It's very bad. It's really a very bad joke that the way they organize this here.
Mm-hmm.
I was in the United States for 6 months. I came back 1 month ago, and it was actually in California. We were much better organized than here.
Mm-hmm.
How is it in Russia?
The situation is very tough now, very tough. I think the best situation just now in China.
Yes, except,
for sure.
Except I cannot go. You see, I work with my students essentially every day.
Yeah.
They are fine. But if I want to go there, they will lock me up for 2 weeks in a hotel room.
Mm.
Hey, they are very strict. And my students are doing fine. But if they want to leave campus, they all have to live on campus. They have rooms on campus. If they want to leave the campus, they have to get 3 stamps from 3 different offices. It's very restricted. So they are not supposed to go into the city even today. Although there is no disease at this moment in Shanghai.
It is in Shanghai.
Yes.
It is in Shanghai.
Yes. So the situation.
Our colleagues, do you remember Kitashov?
Yes.
He is just now in Shenzhen.
Yes.
The situation like in Shanghai.
Yeah,
very strict, a lot of restrictions.
Yes, but it's very strict, but it also seems to be successful.
I think it's effective. Okay. Okay.
Okay, well, I hope things will get better fast in Russia as well as here in Western Europe.
Yeah. Okay.
Okay. Well, it was a pleasure to see you all.
Yeah.
And I look forward to a next meeting.
Yeah. Thank you. See you next meeting.
To the next meeting.
Maybe our students have some questions for Kurt. No?
Sure.
Well, if I may, can I? First of all, hello everyone. I would like to introduce myself. My name is Peter Zaitsev. I'm the secretary of this master program. Actually,
nice to meet you.
Yeah, nice to meet you, Professor Wütrich. Actually, we met the last time you was in Moscow when you were hosted by Professor Kirpichnikov and Dr. Alexei Shaitan. And now I ended up as the secretary on this program, though my research topic is not in the structural biology. So, but the question I would like to ask to you as one of the leading specialists in that area is, well, today we heard the presentations of our students and the head of our program, mostly about structural biology of molecules. But today, I think structural biology is moving forward, and there are some reports about structural biology of even larger objects like cells. For example, I read several papers on the whole cell NMR, and I think that's the more direct way for doing the biotechnology and cellular engineering. So I would like to ask you, what's your opinion of how this, how close is are we towards it, and I mean, how long does it take to do the good resolution whole cell NMR and cellular engineering?
Well, I mean, the trend today is clearly to expand imaging from molecules all the way to intact organisms. And in between, there will be studies of intact cells, and then of multi-cell organisms, or at least parts of organisms. I mean, you may know everywhere now they advertise programs of imaging across scales. And there, you see, when you talk about in-cell NMR, then what is done in these approaches is to overexpress a particular protein and label it isotopically, and then observe interactions of this labeled protein in the living cell with other components. This is not really imaging. I mean, it will give information on mechanisms of interaction of interaction pathways, for example, as a response to signaling by effectors, by ligands, by drugs, by agonists, by antagonists, and so on. When you look at imaging across scales, then single molecule technologies and highest resolution optical microscopy methods are definitely going to give you spatial information on multicellular organisms. And the same, of course, comes from magnetic resonance imaging. You see, and when you then combine in-cell NMR spectroscopy with imaging, there may be a chance to combine again temporal resolution with spatial resolution. This is all happening. This is not just dream. I mean, these are technologies that are in development, actively in development.
Okay, maybe.
Okay, thank you. Yeah, I'm looking forward for it to happen.
That's exciting. It's an exciting world for those who are now 20 years old and start to do their own research.
Any other questions? No? So, so we can finish our meeting now, and I want to one more time express our great pleasure to meet Kurt, to see your health and in so good как настроение? Mood. So this was our first meeting with students, and I hope that we will have such meetings in future. So, Kurt, thank you very much for your time and for attention for us. So,
Thank
Well,
you.
it was a pleasure.
Thank you. It was
Thank
really
you.
a pleasure to see
Okay, all the best, and I look forward to hearing about the progress that will happen during the next few months.
Okay, of course.
Okay, bye-bye.
Bye-bye.
Bye.
Goodbye.
Bye.
Goodbye. Thank you.
Thank
Thank
you.
you, everybody.
Thank you. Goodbye.
Bye-bye.